We are moderating and presenting the session today along with Stroud Water Research Center as part of our mission to advance knowledge and stewardship of freshwater systems through global research, education, and watershed restoration. My name is Mandy Nix, Watershed Education Specialist at the Stroud Water Research Center and member of Watershed Congress Organizing Committee. I'm also your moderator for this session, Patterns and Processes of Fecal Indicator Bacteria in the Delaware River Basin, with Dr. Raven Bier, Savannah River Ecology Lab, and Dr. Jinjun Kan, Stroud Water Research Center. Now, before I turn the session over to our presenters, I have a few quick announcements as we go to our first slide. I wish to thank the myriad organizations and individuals who have made today's program possible. We also recognize and thank our many partners behind, again, what is the first ever virtual Watershed Congress. And for this presentation style session, we will be taking audience questions after the presentation by our speakers. We will be using the question and answer feature as well in the bottom toolbar to take our questions. So if any questions pop into your mind throughout the course of this presentation, please post them there in the Q&A feature in the bottom toolbar. The chat will also be open during the session for comments or technical concerns rather than those questions. I'll be posting guidance for using the chat feature very shortly. So in the meantime, use that to comment and network and make other observations about our session. This presentation is being recorded. Recorded presentations will be made available on the 2020 Watershed Congress playlist on the Delaware Riverkeeper Network's YouTube channel. You will receive an email when presentations from this week are posted there. Finally, this week, we will be using the hashtag Watershed Congress for any social sharing on Twitter or Facebook. But finally, it's time to dive into our presentation, which again is Patterns and Processes of Fecal Indicator Bacteria in the Delaware River Basin, with Dr. Raven Bier, Assistant Research Scientist at the Savannah River Ecology Lab, and Dr. Jinjun Kan, Associate Research Scientist at Stroud Water Research Center. I'm going to turn it over now to Dr. Raven Bier and Dr. Jinjun Kan so they can introduce themselves, as well as their phenomenal research in microbial ecology and biogeochemistry in freshwater systems. Thank you so much.
Okay, well, thank you everyone for joining us today. And thank you, Mandy, for that introduction. So I started off as a research associate at the Stroud Water Research Center in March when this event was originally planned. And I'm now at Savannah River Ecology Lab at University of Georgia, as Mandy mentioned. And Dr. Jinjun Kan, head of microbiology lab at Stroud Water Research Center, really spearheaded this work and will be part of the question and answer session at the end of this presentation. So I want to start off thinking broadly about the overall theme of the Watershed Congress, which is involving the ecological integrity of rivers. So we know that watershed characteristics such as what type of land use the watershed has can directly or indirectly influence the integrity of the river through many aspects such as habitat structure, flow regime, biotic interactions, and energy source, as well as water quality. And the quality of the water can be evaluated by measuring a variety of factors, including how muddy or salty the water is, the water temperature, dissolved oxygen, nutrients, pH, and contaminants. And using these water quality measurements, we can determine if they're too high to maintain the ecological integrity of the water, which can lead us to designate that section of the waterway as impaired. So the dark red areas on this map show the extent of impaired waterways nationwide. And many of these designations occur due to excess sediments, nutrients, or pathogenic microorganisms in the water. And the national average of impaired waterways is 53%, so a lot of states are not in good shape. And the Mid-Atlantic is near that top of the list of concern. However, it's difficult to compare between states due to differences in water densities, as well as different reporting structures for each state. So if we zoom into the middle and lower Delaware River Basin, we continue to see a lot of impaired waterways. But as you look at this map, is often that these data are very limited. So there's a particular challenge in identifying these impaired waterways, and particularly the case for fecal indicator bacteria, which can tell us about pathogenic microorganisms. These fecal indicator bacteria numbers can change a lot over time, and if we don't monitor them with enough frequency, we won't have a good understanding of the baseline values, or if the trend is determining that the waterway is on its way to recovery, or if it's getting worse. So with this presentation, I want to share with you some of the patterns that we've seen for fecal indicator bacteria in the Delaware River Water Basin, as well as some of the processes that may be related to the patterns of occurrence. But before we dive into those research questions, I want to provide some background on microorganisms to boost your familiarity with their features. And I'll briefly discuss their presence and role in watersheds and how they can both cause and inform us about some of the public health concerns. And then I'll jump back into our work on fecal indicator bacteria in the Delaware River Basin with some of the research efforts and where we can go from there. So I'll start off with discussing some of the aspects of microorganisms, including abundance, mass, different types and sizes. So if we think about the word microorganism, this really illustrates the size of the organisms. So mainly we're talking about single-celled prokaryotic, so bacteria and archaea cells, and sometimes we include multicellular and eukaryotic organisms, but overall they're extremely small and too small to be seen unaided in general. So mainly this talk I'll refer to and use bacteria as an example, which you can see at this 1 micrometer size range here. Compare that with the size of red blood cells, which are a little bit bigger, and bigger still are the plant and animal cells. So we know that in general, microbes are quite small. For instance, individual cells, there are different shapes related to these sizes. So we have round cells or cocci, which are about 0.1 to 0.2 micrometers in diameter on average. And we also have some rod-shaped cells, including E. coli and bacillus, which can be on average 0.2 micrometers wide and 1 to 5 micrometers long. And it's interesting to note that bacterial cells grown in the laboratory are usually a little bit bigger than ones in natural environments. And there actually are a few unusually large cells that you can see with your naked eyes. And this includes one called Theomargarita namianensis, which can be 1 to 300 micrometers in diameter, as well as an Epilopsium fischelonsoni, which you can see both of these with your naked eye. And for reference here, here's a tiny E. coli cell. And some of the bacteria cells also form filaments, and some are in sheaths, so lots of different sizes there, as well as a variety of types. We have bacillus with these hair-like pili on the outside, and also a tail or flagella structure here. We have different vibrios, the cocci I mentioned, which are sort of the round ones, and these spiral spirochetes, which you may have heard of. And here we're showing just a diversity of pseudocolors, which can help you distinguish between the different varieties in some of these images. And a lot of these different shapes and sizes can help us classify them with a microscope and also learn about their lifestyles. So currently life is classified into 3 domains. We have the bacteria, archaea, and eukaryota. And humans are part of the animals, which are in the eukaryota. And all of the bacteria and archaea are microbes, and some eukaryotes are also microbes in that very small size range. And based on the current study, bacteria and archaea may have around 90 million species, while eukaryotes only have about 1/10 of that. So there's quite a diversity of the bacteria and archaea. Microbes are also very abundant, and actually the Earth is something we consider a microbial planet. So microbes existed on Earth a billion years ago, way ahead of human, plants, and other animals. So let's take a look at the human population size. So if we consider the size of the human population in September 2020, and we compare that with what's been estimated for microbes, including both soil and water sources, there's about 1.5 times 10 to the 21 microbes for every human. So we're way outnumbered here. If we think about it in terms of biomass, if we look at humans, an average 70 kilogram human comprising 0.05 gigatons of carbon as a total human population, if we compare that with the amount of carbon that microbes have incorporated, microbes outweigh humans by more than 1,500 to 1. So it's quite a number and mass of microorganisms. And that all adds up to show that microbes are the second largest pool of carbon, living carbon after plants, and the largest pool of nitrogen and phosphorus. And if you recall, the carbon, nitrogen, and phosphorus are the most important elements for life. So the human body contains tons and tons of microbes. Also, you may have heard about the Human Microbiome Project, which was an effort to categorize and understand all of these microorganisms on the human body. So we can also, if you consider Earth to be a microbial planet, you can also consider humans to be a microbial body. So we have 37.2 trillion of our own cells, but bacteria cells on our bodies and in our bodies are almost 10 times greater. And where are these microorganisms? So they can be on your skin, in your mouth, your stomach, your intestine, where they're helping you digest. And these bacteria also have distributions on each individual and change over time as you grow from baby to adult, and also change in location. So now I'll start talking a little bit about the significance of microorganisms in natural watersheds. So microorganisms in streams and rivers can occur both in the water column, so these can be free-floating or planktonic forms, and they can also occur in the benthic area, this bottom of the river or stream, and that they can create on these rock or sediment surfaces something called a biofilm, which is kind of like a microbial skin. And it has a very interesting structure. There's some diatoms or photosynthetic organisms and cyanobacteria that occur on the top of the biofilms where they're able to access light. And then towards the bottom of those biofilms, we have the majority of the bacteria and archaea. And this whole film is stuck together with EPS or extracellular polymeric substances. And how do these water samples look under the microscope? So here are some images for stream water, and depending on the stain that you use and the wavelength, you can see what sort of photosynthetic pigments they are. So this image can show some of the photosynthetic cells of cyanobacteria. And then also if you stain all of the DNA and RNA, it sort of looks like a galaxy at night. There's a lot of life here. And these bigger cells are the bacteria, and the tiny dots here are some of the viral particles in the water that people call bacteriophages. And there's also a lot of microorganisms living in soils and sediments. So if we look just beyond the water's edge, upslope to the soil, there's an even greater density. And you can think about when it rains, it's actually washing a lot of those cells into the waterways. And some of the microbes from land can survive and thrive in the water, and some cannot. And in part, this land-water connection is how this happens, and how what happens on the land can influence the quality of the water. So microbes here are in really vast numbers, but what have they been doing for us? In other words, are they good for us? And actually, bacteria have been here for billions of years, providing local and global services. And we're learning that they help us just by maintaining environments. For instance, in 1970, in marine environments, the typical food chain, our understanding of it, started with a small phytoplankton that were consumed by larger and larger organisms up to fish. And then people realized that we're missing a big component here, the dissolved organic matter. So after organisms died, they became decomposed by bacteria, releasing this dissolved organic matter, which is then also taken up by microorganisms and pumped back into the food web. And this pathway is called the microbial loop and can pump about 50% of the total carbon in the ocean. So it's very influential. These microbes are also quite important for transformations of key elements, including carbon, nitrogen, phosphorus, and sulfur. In short, if we don't have microbes, we don't have recycling of nutrients in the world at all. So take nitrogen cycling for an example. Nitrogen gas is abundant in the air and it can only be used by other organisms after nitrogen fixation by microbes or human technology. And so in short, without these microorganisms, there's no full cycle of nutrients occurring on the earth. While many microbes provide benefits, some of the bacteria can make us sick and cause public health problems. For instance, Pseudomonas can cause ear infections at bathing beaches. Clostridium can cause food poisoning. And there are a number of other organisms of concern. In particular, there was a case with Vibrio vulnificus, which caused necrotizing fasciitis in an incident not long ago related to the Chesapeake Bay, where someone was washing a cut with bay water. So to protect public health, we're trying to understand when and where these illnesses happen and what we can do to stop them. For example, the CDC looked at data from 2011 to 2012 and indicated that the outbreaks of illnesses can be associated with recreational water that peaked. These outbreaks peaked in the summer months from June to August. And the collection of these types of data can help us determine how to reduce microbial contaminants to align with goals that were established by the Clean Water Act. So the Clean Water Act is the primary federal law in the US governing water pollution. And the objective is to restore and maintain the chemical, physical, and biological integrity of the nation's waters by preventing pollution sources and providing assistance to publicly owned treatment works for improving wastewater treatment. And it's one of the US's first and most influential modern environmental laws. And it's administered by Environmental Protection Agency along with state governments. And the total maximum daily load is a regulatory term for the US Clean Water Act describing what is the maximum amount of a pollutant that a water body can receive and still meet the water quality standards. So they've been used extensively by Environmental Protection Agency and state agencies, including evaluating nutrients and sediments, as well as microbial contaminants, where those are ranked as the #1 for causes of water quality degradation. And this can lead to impairment of many, be associated with impairment of many rivers and streams, lakes, reservoirs, ponds, and a lot of the assessed bays and estuaries. And as an example, let's look at the Brandywine-Christina Basin, which is one of the 8 subclusters within the Delaware River Watershed. And based on the EPA bacterial TMDL for fecal coliform, the majority of waterways in the basin were classified as impaired, shown here in red. But keep in mind, some of the areas upstream in the Brandywine were not classified as impaired, which is not necessarily because they're in good shape, but could be a lot of times because we're lacking data. So how do we evaluate bacteria in water bodies to determine if they are impaired? Well, pathogenic microbes are often associated with fecal waste and can cause numerous diseases, but it's difficult to measure directly. It's both expensive and impractical. So we instead use a group of indicator bacteria that are fecal associated. So these are coliforms, E. coli, and enterococci. And to be a good indicator, these groups should meet the following criteria. They should be present when the pathogens are present and vice versa. They should be more numerous than the pathogen, easier to cultivate in the lab, and they should be more resistant to die off in the area of the water that's being tested. So some options for bacteria monitoring are the total and fecal coliforms. And within those groups, the E. coli and a subset of these E. coli are known to be pathogenic. Another group to measure includes the Enterococcus genus, with many members, some of which can cause illness. And so out of these groups, which are the best ones to monitor? Well, it depends on what is the major route of exposure, whether it's consumption or recreation. For recreation in freshwater, the best indicators are E. coli and enterococci. In saltwater, the enterococci are the best indicators. And actually, until 2004, fecal coliforms were used also to indicate the safety of saltwater beach recreation. But for now, for recreational waters now, total coliforms are no longer the recommended go-to as an indicator. And for water supplies and standards, the broader group of total or fecal coliforms are still used. For drinking water, total coliforms are still a standard test, in part because the presence can indicate contamination from an outside source. So as we've learned more about bacteria monitoring, we've been able to identify more specific and accurate indicators, and this effort is ongoing. So what bacteria have been monitored historically since the 1920s? Public health agencies have used total and fecal coliforms. There can be a non-fecal origin of these bacterial groups, as well as having coliforms that are regrowing in natural environments. And they're still being used by many state agencies, particularly for drinking water, as I mentioned. Now, the E. coli and enterococci groups are more specific bacterial groups. They're currently the most commonly used and recommended by the EPA. So a lot of this effort to determine what to measure requires looking at the relationship between indicators, pathogens, and illnesses. So from a 2018 study, there was an evaluation of 45 such documented relationships between indicators and pathogens for freshwater, shown in the green. Yet it's much more difficult to link these occurrences with illness. And part of this link differs by the technology that's used to assess the bacterial indicator. So EPA is reevaluating these approaches, and their 2018 review of water quality standards, they found that the Enterococcus species measured by qPCR might be more predictive of GI illnesses. And so these approaches are consistently undergoing reevaluation and changes. So currently for recreational contact, we need to evaluate samples over a 30-day period at base flow, so when the water is not influenced by rainstorms. And the current acceptable level of contamination should keep illnesses to no more than 36 per 1,000. And using enterococcus as an indicator, this threshold is fairly low at 35 for a geometric mean, whereas for E. coli, it's at 126 for fresh waters. And this is the Pennsylvania DEP recommendation for recreational contact during the summer season. So to conduct these measurements at Stroud Water Research Center, we use an IDEX package where we mix the stream water sample with a food that contains a chromogen that reacts with an enzyme found in total coliforms, or a fluorogen reacting with enzymes found in E. coli. And then we look for changes in color or UV fluorescence to indicate that the fecal bacteria are present. And then based on the number of positive and negative wells, we're able to use a statistical approach to determine the most probable number of colony-forming units for that bacterial group. So now I will get into our research questions that we address using some of these techniques. So I want to look at some work we've done to understand what are the spatial and temporal patterns of fecal indicator bacteria in the Delaware River headwaters, which environmental factors can best explain the presence of these bacteria in headwaters, and what the macroinvertebrate aggregated index for streams, how that score of different site quality can correspond with the abundance of fecal indicator bacteria, as well as the entire community, and how well non-fecal indicator bacteria can correspond with those macroinvertebrate water quality scores. So to address these questions, we sampled different sites throughout the Delaware River water basin, with a focus on the Brandywine and Christina watersheds, but reaching some sites further north as well. For our field sampling design, we focused on the headwaters within these different watersheds. And we mainly sampled during the summer months when counts and recreational contact are shown to be the highest, so June through September. And we sampled different frequencies, bi-weekly, monthly, and over multiple years. So I'll start with one of our first research questions. What are the spatial and temporal patterns of the fecal indicator bacteria in those headwaters? So here are some graphs showing the spatial data for summer E. coli abundance at sites across these headwaters from 2016 to 2019. And you can see that the majority of the highest values of E. coli are occurring at a set of headwater sites, often in the Brandywine and Christina areas shown in blue, as well as the middle Schuylkill shown in green. And these have also been documented with very high numbers. So most sites are well over the 126 threshold that's recommended by EPA. And if we look at the abundance of enterococci species for these sites and years, we can also see spatial variability within and across watersheds. But again, many of these sites are exceeding the threshold for enterococci, which is set at 35. So when we focus on a subset of 4 headwater sites in the Brandywine-Christina watershed, shown here, we can also identify seasonal trends. So these graphs are for E. coli from these sites from 2017 through 2020. And the time is designated by the month and then the week within that month of sampling. And there are many high value occurrences happening during these summer months. But in nearly all months, one or more of the sites was exceeding the 126 threshold, shown with the line in red. And enterococcus at these sites also shows a similar story, with spikes in abundance occurring throughout the year, and particularly in the summer months as well. And again, over the threshold shown in red. So we're seeing these high fecal indicator bacteria values for headwaters, and next we wanted to try to understand what environmental factors might be most closely related to them. So we looked for we ran through a number of exploratory analyses using correlations, and we looked at watershed size and different types of land cover. So when we looked at watershed size as an explanation for the abundance of E. coli, we saw no significant relationship. So both small and large watersheds had both high and low values of E. coli abundance. For enterococci, however, we did see a significant relationship, and in fact, as watershed area increases, the abundance of enterococci was decreasing, so large watersheds have fewer enterococci. And in addition to watershed size, we did identify some weak correlations between types of land cover and either E. coli or enterococci abundance. There was a significant negative relationship between forest cover and E. coli, as well as for both fecal indicator and the density of structures, roads, and other impervious surfaces, as well as open water. And we found some weak positive correlations occurring only with E. coli between the percent of low vegetation, as well as agriculture and grass, which can include lawns, pastures, and shrubs, nursery plantings, etc. So here are the graphs to show those relationships, starting with E. coli and enterococci with the percent of forest cover. So again, E. coli was the only group that had a significant relationship, but it was a fairly weak correlation and slightly negative with the increasing percent of forest cover. And when we look at the percent of agriculture and grass within the watershed, again, enterococci was not significant, but we did see a weak relationship with the abundance of E. coli and the percent of agriculture or grass. So there's been a lot of work on using macroinvertebrate communities, including aquatic insects, as indicators of water quality. And so we asked if the stream health index values that are based on macroinvertebrate scores could correspond with fecal indicator bacteria abundance, as well as the composition of the full community of bacteria. So for this study, we used a subset of sites spanning a good, fair, and poor quality based on the macroinvertebrate aggregated index for stream score or MAIS score. So you can see the different colors of those ratings shown here in blue for good, yellow fair, and red for poor. And at each of the MAIS quality scores, whether it be good quality shown here in blue or poor quality shown here in red, we found that the abundance of E. coli, of fecal indicator bacteria, was exceeding that EPA threshold at the majority of sites across multiple years of study from 2016 to 2019. And further, there was not a trend between the MAIS score category and the abundance of E. coli. So one site here does have a very high abundance, but some of these other poor quality sites are much lower compared with the fair and even good quality sites. And we found a similar lack of trend over space or time for the enterococci abundance in water at these sites. So next we wanted to see if this trend could be more apparent for the different habitat compartments and for the full bacteria community. So this graph shows bacteria samples collected at the MAIS sites from water sources, rock sources, or sediment sources. And each point represents a community of bacteria from that type of sample. And the closer together the points are, the more those communities share in common, including their identity and the abundance of each organism. And the further apart they are, the less they share in common. So when we compared the bacteria community composition between habitat compartments, we found that each habitat had a distinct bacteria community. When we aggregated the communities from all the different habitat types together, we found that there was a difference between good and poor type MAIS scores, as well as between fair and poor, but no difference distinguishing between good and fair MAIS score communities. And we also found if you evaluate each of these habitat types independently, there was no difference that we identified between the MAIS site types. So this suggests that to differentiate between these site quality categories, we need to know bacteria communities for more than one habitat compartment. So last, I want to look at how well non-fecal indicator bacteria correspond with these MAIS scores. So we wanted to determine if other bacteria in the community could help indicate the quality of a site other than the fecal indicator bacteria. And we identified some other indicator groups that correspond with a good quality or a poor quality site. And these are listed here. But there are very few that indicated fair quality sites. So there's ongoing work to evaluate some of these groups, especially the bacteroides, which could be helping us identify some of the poor quality determinations in the future. So now I want to briefly discuss some of the work by Dr. Kan and others that seeks to use what we know about microbial communities from different sources to identify where fecal contamination is coming from. Some of those sources include wastewater treatment plants, septic systems, as well as humans, livestock, pets, and stormwater runoff. So this is a type of microbial source tracking that can help us identify these contaminant sources by matching the gut microbiome of the host with the contaminants identified in the waterway. And there are lots of different choices for source tracking methods. So we use their number used at Stroud Water Research Center. And I'll just briefly go through some work that was done on primers targeting bacteroides, bacteria that are specific to each host organism resident bacteroides. So for this study, they used 11 sites in the White Clay, Red Clay, and Brandywine creeks. And they found that throughout these different sites, the White Clay, Red Clay, and Brandywine, there were different sources associated with humans, cows, and all bovine, shown in the different colors here. And this was most clearly shown in the Brandywine for some human sources, as well as some sites with cows and deer maybe contributing the most contaminants. Now I just want to briefly summarize the work that I went through for the fecal indicator bacteria in the Delaware River Basin. So we found that of the observed sites, most of the sites had E. coli and enterococci concentrations that were greatly exceeding the EPA thresholds. The enterococci concentrations were the highest in headwater streams, but E. coli had no watershed area relationship. There was a weak relationship with watershed size and land covers, but mixed sources could confound these patterns. And we also found that even good MAIS scores for the macroinvertebrate scores exceeded the EPA thresholds, and some similarities were identified in the scores and the full bacteria communities. And we're also working on some source tracking and some potential for identifying sources of contamination. So some future perspectives, there are a number of questions remaining for helping us identify why these patterns of fecal indicator bacteria are occurring and what we can do about them. So specifically, we can ask why are those counts so high for E. coli and enterococci during the summer? Where are they coming from? What's causing the high variations? Is it temperature, land uses, local sources, different wildlife? How long can the fecal indicator bacteria survive or travel through the watershed? Are E. coli and enterococci good indicators for headwaters? Should we use the full microbiome in the water column and on the stream beds? So I want to say that a key for this is continuous monitoring and more work. There's a lot more to be learned about these environments. And so I want to also acknowledge many of the contributors to this work listed here, as well as the funding organizations, and also a large thank you to the organizers, including Mandy Nix, Sherry Town, Scott Ensign, and many others for doing all of the hard work to move this event online. And with that, I will stop the presentation here and take any questions you might have.
Thank you greatly, Raven and Jinjun. This was greatly helpful and informative just to get more perspective on microbial ecology, which we rarely get in our freshwater sector sometimes. And we do have our first question today. It can be answered by either Raven or Jinjun, based on who's comfortable, or both. This first question is coming from a participant who wants to know, can you characterize the land use around sites with higher levels of ovine and bovine and those with higher human bacteria? Again, that question is, can you characterize our land use around sites with higher levels of ovine and bovine and those with higher human bacteria?
I think that some of the microbial source tracking work is starting to evaluate that and show some of the relationships between different types of agricultural land use and the contaminants that might be associated with some of those sources, depending on whether you're perhaps fertilizing or having livestock in those land areas. Jinjun, I don't know if you want to talk a little bit more about some of that source tracking and introduce yourself as well.
Sure. Yeah, sorry I joined the webinar late. Have another meeting. Yeah, I think what Raven said is correct, and we are still. I mean, the source tracking is really a very complex issue. You know, the microbes are so small you cannot easily identify them even with the microscope. So we have to rely on this DNA approach. When we have different kinds of source materials in this mixture, right? You're really looking at the water is a mixture of the microbes from all over the place. And identify each microbe, where they're coming from, and the identity of those guys are not easy. So that's why they come up with different kind of approaches here. And the Raven showed the data here is called a quantitative PCR. It's a really sensitive approach. And also we are thinking about in these days by using the high throughput sequencing, which is rely on the total community structure, hopefully that can provide us a higher resolution level of identification. And hopefully we can get a better, get closer to the reality or to the truth, what can identify what microbes really coming from the cow, from the human, or from any others. So any land use, just keep in mind, we monitor this headwaters. However, the water can come up from different land uses, right? And they depends on the time, depends on the location you sample, you may deal with different kinds of a mixture. And how you partition those sources, identify those source materials is really a big challenge, even with nowadays with the current technology.
Fantastic. Thank you, Raven and Jinjun. We have another great question in the question and answer feature. It's actually a two-part question, so I might tackle the first with you. And it is about whether some of the bacteria come from the rain or specific south to northerly flow of storm systems. Again, could some of the bacteria come from the rain, or could it come from specific south to northerly flow of storm systems? What are our thoughts?
Okay, I will take the first crack. Raven, you can feel free to add. And this is not the first question that I was asked about this, and that's a really interesting phenomenon actually. The really storm event can bring in the microbes from the wind or the cloud, the rain, right? And the reality is even for the stream water, thinking about you have about 1 million cells per milliliter. So if you know 1 milliliter of stream water, you may have 1 million cells. And compared to the stream water or any other surface water, the rainfall, they bring in those microbes. Compared to those, they're really minor. I mean, we're talking about maybe less than 0.1 or 0.01%. So to get back to the question here, I think it's possible those microbes will be mixed in our stream water, but I would say the proportion of those microbes will be very, very low. And the chance of those microbes become a contamination sources will be really very low and very, very rare.
Fantastic. Raven, I don't know if you have any thoughts on that before I let you know what the second part of this question is, which relates greatly to what Jinjun was just discussing, which is it possible that the lack of a presence of a larger bacteria, which may feed on other bacteria, could be a factor? Is it possible the lack of a presence of a larger bacteria, which may feed on other bacteria, would be a factor?
I think mostly I'm thinking actually about smaller sources of mortality for bacteria, including viruses, which we've started to learn a bit more in terms of bacteria mortality and the high turnover of bacteria cells, some viruses that are very specific to different types of bacteria, or some are more general. And we're really just starting to learn about that source of mortality. As far as I understand, I think it's quite a bit greater than maybe larger bacteria cells for that mortality. But I think that would be a great question in terms of sort of more of the ecology of how the fecal indicator bacteria is surviving in these waterways over time.
Yeah, something I want to add here, Mandy, is this is a great question. And I think based on my knowledge, I'm not aware of any bacterial cells feeding on the others because they don't have that capability, just because they are not, as a human, they don't have that toolbox to do that. However, let's put it this way, the microbes, they do interact with each other. They're not feeding on each other. However, give you an example, I mean, this is pretty new. I think University of Delaware, there's one lab down there. They found these two microbes. They're different kinds, okay? They're different kinds of microbes, but they can fuse together. After they fusion together, they become new microbes. I mean, that's just to give you one idea. The microbes can do this kind of stuff. And also for the fecal indicator bacteria, I assume that's the question asking about, right? That's a pretty factor in here. But the fecal indicator bacteria, they probably cannot rely, well, they don't rely on the others, or they won't feed themselves to the others. However, after they are released into the environment, they will exist there. They probably can survive that environment. And then you're thinking about the environment conditions can be harsh, right? Sometimes, and a lot of microbes already in there. And then we're talking about these microbes, the interaction between microbes. Some microbes can get along. So those, they may compete for some certain things, right? So that's one big hypothesis. I mean, kind of follow on Raven's talk here. We really, this is a big puzzle, right? We try to understand why you have these high numbers of bacteria in the headwaters. And one hypothesis out there is, and I believe that hypothesis is just because the E. coli or enterococcus, they are out there in the environments, and when the favorable conditions come, for example, in the summertime, the water temperature is higher, and then it just suddenly become like, become more actively involved, become more reproductive. So that's one hypothesis. I mean, back to the question, computation is another way, right? The microbes may compete for certain sources like nutrients, right? And of course, the microbes, they cannot eat other microbes, but they rely on a lot of nutrients, a lot of carbon sources. I mean, so they rely on those things, and the microbes really, they compete to each other and they try to survive. So that kind of interactions are always out there. And those are very important factors determining how this microbial community exists, coexist, and survive.
Brilliant. Thank you, Jinjun. Thank you, Raven. I think what's clear here, like you said, Jinjun, wonderfully, is this is a puzzle. And it's both deeply challenging work, but profoundly important work, not only for human health, that is clear for most when we talk about things like E. coli, but also for watershed health. And so I think for now that may conclude some questions, but let's give folks a few more minutes just to see. We've got until roughly 3:30 on this session. If anyone else feels that Jinjun or Raven, as they expertly navigated those questions, provoked something interesting or exciting to them that they want to know more about, pop into the chat or the Q&A feature at the bottom toolbar and let us know if you have anything else to ask our experts here with microbial ecology, as it relates to fresh water. But
while we're waiting for more questions, Mandy, I think after this presentation, Raven did a good job explaining our data. And I think one question we don't want to scare all the people away, say, wow, okay, the headwaters are so many bacteria. Let me tell you this. I mean, no matter what, you have microbes out there in your stream. Let's say, I think a lot of the audience probably thinking about, geez, I have a house, there's a stream back in my house. Is that safe for me to go in there? I think that kind of question, I get a lot of those questions in these days. I guess the short answer is yes, you should take a precaution in summertime, especially because the E. coli and enterococcus, those two different types of bacteria, they are just an indicator, right? And the worst case scenario is those microbes may come from human. You don't want that happen because that means some sewage plant or sewage facilities may be down or something, so you may have that contamination issue in your streams. And then along with those, I mean, the pathogen, so enterococcus and the E. coli, most of them are not pathogenic, okay? Keep that in mind. So we're not monitoring the pathogens, we're monitoring the indicator bacteria species. So just when you have that high number of indicator species, that's just to give you a warning, say, oh, I may have some issues in my water. But on the same time, flip side is you may have those bacteria in your background, in the environment already. And a lot of studies have done to show the E. coli, for example, they can survive the natural environments for a long time. So with that being said, I mean, yeah, sure, you take precaution, especially when you go to, do something in the stream water and take precaution. Meantime, don't scare yourself, don't be freaked out. And also Raven showed the bacterial standards, right? Those are from the EPA, most time for drinking water, for recreational water. And, really we're talking about the contact level here. You're not really dig yourself into the water, bury yourself in the water. That's not a full body contact, right? So with that being said, the risk out there is not that high. And we are still trying to figure out, that's why the science, we're not moving fast enough, I guess, because we need a lot of data. That's why Raven mentioned about last slide, we need to continue monitoring this, try to understand this, try to identify the source materials, like where this bacteria coming from. So to answer that question is not easy, but I mean, that's my suggestion. Take a precaution, but don't be freaked out. I mean, this just indicate the bacteria, and they may just give you some hints about, your background, your backyard stream, and how good or how bad the stream health is.
Perfect. Thank you, Jinjun. That was very helpful expounding on that, because again, this is really the intersection of human and watershed health here. With a lot of unknowns that are powerful, but they shouldn't make us scared. And with that said, there's actually 2 questions now in the chat. We're going to answer just these 2. They'll be our last ones for the day before I close with a few statements. But these questions, actually, you might have time for 3 more. The first one that relates back to what you were just saying, Jinjun, about these unknowns as we're looking for more information is about microplastics. And we have a lot of microbial colonization of microplastics, but microbial diversity absorbed in microplastics has been noted to be significantly different than that in the water column and suspended organic matter. Is this an emerging concern in your field? And I would answer this briefly, so we squeeze in maybe 1 more question after this. But what are our thoughts on microplastics and microbial colonization of them as a concern in our field?
Go ahead, Raven.
Okay, I was just going to say, yes, this is absolutely an area of interest and very rapidly growing research area. And really, we don't know yet what these differences mean. Absolutely, we're seeing differences in the diversity colonizing those microplastic particles, more microplastic particles, and what is the fate of those. One question might be how the diversity of organic matter particles that might be incorporated into some of those higher trophic level guts might be changing if microplastic microbial diversity is being ingested instead. So yes, it's absolutely a place of interest.
Fantastic. And I have a great question in here too about indicators. Jinjun Kan mentioned indicator species that we can often see in bacteria that are some of the first indicators there might be a problem. They can be the most sensitive to pollution and other habitat disturbances. This question says, I might have missed the macroinvertebrate community indicator correlation with E. coli. Did you find a strong connection or correlation between macroinvertebrate indicators and micro indicators?
So we actually did not see a strong connection compared to what we were expecting. We had categorized a good, fair, and poor site quality based on the macroinvertebrates, and actually found that at all of those sites, there was not only an exceedance of the threshold for E. coli in those waterways, but also that there was not at least a linear trend over the different years that we examined for more E. coli being present at the poor quality sites.
Brilliant. And we have time for one last question before a few closing statements and reminders. This is from a participant who would like to know, if there is not a specific known source of bacteria problems, what is your concern on the level of bacteria? And is there any way to limit or protect the public other than staying out of the water?
Well, this is a good question. I mean, yeah, a lot of our samples actually we cannot identify that, always the sources, right? I mean, we do have a couple positive control sites. Say, okay, this is the cow, and this is a septic system. We have a field sewage facility or something like that. So we have those positive control. But to be honest, when you go out there, a lot of times you probably don't know the known sources, right? So, I mean, let me back up a little bit. I mean, I think Raven mentioned about how abundant the microbes are in our water. Actually, 99% of them are good bacteria, put this way, right? So we really benefit from those big numbers. I mean, thinking about the leaf fall, right? I mean, where the leaf fall goes, if you don't have microbes, they probably get accumulated until we get buried about those. So remind me again, Manny, what's the question? Sorry, I lose the track a little bit. I mean,
yes. So they're wondering, from the public perspective as well, is there any way to limit or protect the public other than staying out of the water? And as a reminder, recommendations for that, Raven and Jinjun, are roughly 48 to 72 hours after major stormwater. But what recommendations like that and others can we give to the public?
We don't have that, recommended. Well, that's the reason why, we're working on this, right? So really, the biggest question to me, to my lab is like, okay, in summertime, we notice this high numbers of indicator bacteria. What does that mean? Does that mean the water is not safe anymore? You cannot go in there? And Mandy, I think you made a good comments on this, like after storm event. Usually, I don't suggest you to go out there. I mean, of course, safety, that's one concern. Another one is we're talking about a couple of different things here. You may have the sewage, right, failure. You may have the wastewater management. But no matter what, when you have a storm event, you always have that high numbers of bacteria in the water, because the bacteria always be associated with the particles. So when you have a high particle concentration, you always have high, microbial cells in the water. So I would suggest, you definitely don't want to go in there right after storm, after 48 hours. Yeah, you feel like a better, but I think that's not the key. The key is to try to understand more about the microbes, about the indicator bacteria species in the water. That's more important. It's not like, the number is lower and I can go out there with no problem. No, that's not that's why it's, the EPA try to set up these standards are not for, a lot of times you probably notice that each state has their own standards for drinking water, for example. That's more critical. Or a swimming pool, if you cannot really use those standards for our headwaters, as I said, we're really talking about different kinds of contact levels, right? We're not drinking the water directly from the stream. So, I mean, I guess all those things put together, well, back to the question, the general answer is no, we don't have that kind of guideline. However, I mean, that's why we're working on this, right? I mean, hopefully we can provide the EPA, provide the stakeholder or the policy manager and say, okay, we recommend, this. I mean, even in the swimming pools, and if you have the one number go over the limit, and that's a red flag, and then the technician needs to come back and resample the water to confirm these results before we recommend any closure. So, that kind of a protocol will start in place and do those kind of, but for the headwaters, we don't have that kind of a policy right now.
Thank you, Jinjun. You and Raven, I think, have masterfully articulated why this is such important work. Again, that is at the intersection of watershed and human health, and we should not be fearful of these bacteria. Most of them are good, but we should be in support of more research like yours that can help us better understand them. So I am afraid it's 3:32. It is time to wrap up our sessions today, but I want to take just one more moment to thank our speakers once again for taking the time to share their research with us during this week of the first ever virtual Watershed Congress. Also, we encourage you to virtually tour the Watershed Congress's virtual exhibit hall. All in one place, you can find more information on Stroud Water Research Center and other supporters of the Watershed Congress, as well as fun opportunities to connect with them and discover resources for your work. I've posted the link for that in the chat today. And as a reminder, this session was recorded, so you can watch it back later on the Delaware Riverkeepers Network YouTube channel. It should be going up in the coming days to weeks. And finally, we hope this session sparked new and old enthusiasm for freshwater research that will continue to burn brightly throughout this week's Watershed Congress sessions. But check your email and calendar for additional session invites like this one, and we hope to virtually bump into you again at another session this week. So big thank you, everyone. All right, have a great rest of your Tuesdays. Bye.